Thank you. It's always a pleasure to talk to those people who have been so interested in our work for so long. Um, and I have to say, I am really excited and get more excited every day about the progress you made. So I'm just going to take a minute to talk to you about what I think the progress is that we have made. Specifically, we now actually can measure Alzheimer's disease in life. And so I want to point out that what a huge advance that is. We now are able to determine if someone has amyloid in their brain by many different ways, and we can determine for sure that they have Alzheimer's disease. We don't have to use that term probable Alzheimer's disease anymore. And knowing that, we have been able to identify very specific cohorts of people who have the disease and determine that we can actually have a clinical benefit by some of the new anti-amyloid drugs. So I want to make a comment that, in fact, amyloid is not a hypothesis. It is a definition of the disease. And many times you'll be hearing some studies where we might be measuring amyloid, but it's to determine if we're collecting people who have Alzheimer's disease and not some other related dementia. So I just wanted to remind you about what importance we have in our AD biomarker pathology. So we know that we can measure amyloid deposition. We can do it with PET imaging and CSF, and now we can do it pretty reliably with blood or plasma. We also know that tau, which is the second pathology that's also related to Alzheimer's disease, can be measured PET and CSF and some measures in blood and plasma. And then we also know that staging the disease occurs by measuring the neurodegeneration or the neural injury. We can do that with MRI or FDG PET, and we now have some blood biomarkers that tell us about the severity of the disease or the severity of the neurodegeneration. So as you can see, the amyloid plus the tau are specific to telling us that it's Alzheimer's disease, and the N, the neurodegeneration, that's for the staging. And so what you're going to hear about today is a series of phase 2 trials that are examining different interventions. None of them are directly amyloid, and they are selecting individuals who have Alzheimer's disease and seeing how we might modify their course. So I want to begin with something called the START study, and it stands for Synaptic Therapy of Alzheimer's Research Trial. And the drug is so new, it's a phase 2 study, that what we see is that you have only a number, right? So this drug, C21812, is a drug that is a small molecule, which means it can cross the blood-brain barrier, and it doesn't need to be administered intravenously. It can be administered orally. Now, what do we know about this drug? Well, this drug blocks the binding of oligomers, right? Our anti-amyloid drugs right now actually are monoclonal antibodies, but this one blocks the oligomers, which are the pre-stage to amyloid. So this idea of blocking oligomers is important both for people who have amyloid and possibly for people who've had it removed, but they still have amyloid oligomers accumulating. The idea is that it will restore the function in the synapse, which is the connection between neurons that allows us to actually learn and do our cognitive activities. So you see here, I have a picture of our coordinator. This is Trude Silberstein, and she is the coordinator for this study. You may know her. She's been with us for a while, but I wanted you to have a familiarity with some of these people so that you can know who you're speaking to when you make your contacts with us. So let me tell you a little bit about the START study. It's got a wide range of 50 to 85, but it's for individuals who have mild cognitive impairment or Alzheimer's disease, dementia. So what does that mean? That means that they have symptoms. There's a memory deficit that's truly apparent. And we know that it's not just the silent amyloid, but that they have symptoms. They can be mild symptoms. In fact, they can be mild cognitive impairment or mild AD, but they do have symptoms. In addition, for eligibility, you have to understand that this is a long study. It's 20 months time commitment. And the requires you to take an investigational oral treatment, which is certainly has some simplification over the use of a monoclonal antibody, which currently needs infusion. And as with many of our studies, we have to have a study partner. In fact, you'll see this on the slides, and this is a requirement for really all of our studies because we need a co-partner to help us be sure that the medication is being delivered, that the reporting of side effects are being complete and thorough. So this slide might be a little hard to read, but I just wanted to give you some broad aspects of what this study is about. This study begins with several screening visits, up to 5 visits. As some of you who have participated in our studies know, screening can take a while. There are things that have to be done in a certain order. We have to confirm the safety for individual patients, confirm what medications you're on, confirm the presence of amyloid, confirm what other side effects or history you might have. So this study will examine both the presence of amyloid. It will assess the presence of apolipoprotein, E4 allele. It will also assess all your medical conditions. This is followed by randomization, which starts with visit 5. This is when you qualify for the study and you are randomly assigned to receive either drug or placebo. But in this case, there's actually 3 choices because there's 2 doses of drug, a dose of 100 milligrams and a dose of 200 milligrams, as well as the placebo. And the medication is taken in capsule form. And as you know, placebo is designed to look identical to the treatment. So the treatment period includes visits from 6 to 23. And in these visits, you will be taking study drug, attending in person for medication counting and medical tests, as well as our outcome measures and procedures. And then finally, there's a follow-up phone call that'll occur at the end of the study. So we tell you this so you'll understand the burden of the study. Now I want to tell you about another drug trial that we're doing. The trial's referred to as benfotiine, and the drug is something called benfotiamine. This is a very interesting drug. It's what we call a prodrug to thiamine, which has a very high safety record. It's almost available in some doses in the health food stores. The problem is you can't get high enough levels of thiamine to see the kind of effects that we're hoping to see. So by using benfotiamine, it's an inactive compound that becomes the active compound once it's in the body. And it's absorbed better than thiamine, giving you higher levels. Now, many of the properties raising thiamine in the blood are the reason why we use a prodrug in this case. Now, there's a lot of discussion that we are only testing amyloid drugs, and I would argue that benfotiamine is a really good example of not targeting amyloid specifically. Really, it targets the strengthening of the neuron and the strengthening of the synapse. And we say that it has anti-inflammatory and antioxidant effects. So in benfotiamine, there are some entry criteria. And again, I wanted you to see who is our coordinator for this study. It's Joanne Lim, who's been with us for quite a while, and she's the major coordinator, but I will explain to you that all of our coordinators can take part in any of our studies to make sure that we have coverage for all of the times that a patient is coming in. This study is specifically for people aged 50 to 89 experiencing the same type of MCI or mild AD. We want to confirm that a person has Alzheimer's disease. So in this case, we're using a blood test to confirm that. The requirements here are simply to be stable on current acetylcholinesterase inhibitors. That is drugs like drugs like galantamine and donepezil, which are currently used for almost any type of memory loss. Individuals can be living in the community, and they have to be, again, willing to participate in this study for up to 18 months. So what about that screening process? Well, the screening process for benfotiamine also includes a physical and cognitive testing and MRI scans and blood tests. It's not dependent on an amyloid scan because amyloid is being evaluated through the blood work that we will be doing. So some additional details about this study. It's an 18-month study, and it requires, after screening, individuals are seen once a month for 3 months. Once we know you're stable, then our visits go out to every 3 months. So that, again, reduces some of the burden. So what happens during the visits? There's blood testing, there's imaging, and there's cognitive testing as well. And our clinical trials are offering compensation. This one in particular is offering $100 per visit to both the participant and the study partner. So the study partner is very critical to us. And what people often ask us is, what information will I get back? And in the case of this study, we will provide you with the results of your plasma blood test for amyloid, and we'll tell you whether or not it's positive or negative enough for you to join the study. So while we won't give you an absolute value, you can use this to determine whether or not it's likely that you have amyloid and therefore likely that you have Alzheimer's disease, or perhaps not. Another study that I want to tell you about is a study called Tau RX. This is a study that is sponsored by Bristol Myers Squibb. And again, this is a phase 2 study, so the drug doesn't have a name yet. It has a number. And the PI of this study is Dr. Soleimani. Many of you may know her already. And we have, as our study coordinator, the lead coordinator, Ame Aisher, and she is one of the people that you would contact about the study. So what is Tau RX? How does this study work? So this is an anti-MBTR monoclonal antibody, right? So we know a little bit about monoclonal antibodies. Those are the things like Leqembi and Consuela, the most recent monoclonal antibody for amyloid. This one is a monoclonal antibody for tau. And the theory is that the accumulation of soluble tau plays a key role in the disease progression. So the antibody binds to MTBR to reduce the spread of extracellular tau and to prevent it from getting inside the neuron. So theoretically, it would potentially slow the progress. You can see this is, again, a non-amyloid mechanism that we're studying, and that using this same monoclonal approach, which we know has worked with other proteins, amyloid in particular. And the study is um, particularly important in that it's one of the first well-designed studies going forward that will actually approach tau in the brain. You remember my very first slide, I talked about how amyloid was important and tau is important. And tau in particular is important to know where it accumulates in the brain. And that's why the imaging is so important in this study. So what about the screening here? Well, eligibility is dependent on age, of course, 50 to 80. Individuals who have MCI, mild cognitive impairment, due to Alzheimer's disease or mild AD. And so what does that mean? If we're saying it's due to Alzheimer's disease, we have to confirm that, and we confirm that by the evidence of tau and amyloid from either an LP or a PET. Most of our patients prefer to use the PET, but the LP is available. Individuals can range in their mini-mental score from 22 to 30. And they have to be on stable medications. And for this study, individuals cannot be on anti-amyloid treatments, right? So that would mean that an individual who is currently taking one of the approved medications would not be eligible for this study. We also require, as I mentioned, every study requires a study partner. And again, here we have the requirement of a study partner. So some more details. How long? It's 2 years after screening. And how often? Well, after screening, it's monthly. And at those visits, things that occur include infusions and blood draws, imaging and testing, including cognitive testing, as well as in some cases, other physical testing. And this study, again, does offer compensation. The participant is offered $150 per visit. And what's returned to you? Well, again, the details that are returned are the blood test results in terms of whether or not you have elevated tau, p-tau, which is actually a measure of amyloid, as well as elevated tau. So these blood results will determine whether or not you're eligible to even go forward in the study. And if you're not, it's probably due to the fact that these biomarkers are low, meaning that you're unlikely to have true Alzheimer's disease. So finally, I'm going to be telling you about one of the studies that we've been doing for decades now. That's the ADNI studies. The ADNI studies have been going on here at Mount Sinai since they first began more than 20 years ago. And they are one of the most important and productive studies. They have provided data sets that have been used by many investigators and researchers around the world. And the ADNI studies have really helped us design the best clinical trials. So for individuals who are not ready to consider a clinical trial, an observational study like ADNI might be of value. So what do you need to know about these studies? Well, as I say, they're the longest running studies that I know of in Alzheimer's disease. They're observational, which means we're not treating, and we're data collecting, but we're data collecting in a very precise way. And so eligibility is often as detailed as it is for a clinical trial. Our coordinator, our main coordinator for this is Joanne Lim. And I think what's interesting is that we can recruit a wide range of individuals, but again, they must meet some of our criteria. So normal controls, mild cognitive impairment, and dementia. And what the asterisk is there for is to say that for the mild cognitive impairment and the dementia, we include in our screening visits confirmation that there is truly cognitive impairment and not simply cognitive complaint. The age range is the highest of our studies, which I think is so important because 85 and 90-year-olds are now not uncommon, and we want to make sure we know what's going on with them and what their trajectory of cognitive health is. So the overall purpose of the study is to understand Alzheimer's disease and related dementias with new biomarkers and new tests. It's very exciting. The um, the ADNI data has really helped us understand what we expect to occur over time so that we can assure that we know what the trajectory is and plan our trials correctly. So this commitment is for 5 years, and it's only once a year, although sometimes it takes several visits in that once a year period because we have so many things that we're measuring. There is compensation both for the study partner and the participant, depending on which procedures are being done. So in terms of returning results to you, any FDA-approved test, such as amyloid imaging, it is returned, the results, if you want them. There are some people who choose not to take those results in. So this is an option for individuals who are curious about the latest biomarkers, wanting to know their status, but not ready to make a conviction, a commitment to a clinical trial. So I wanted to just remind everybody that, you know, while I've shown you some of our participants and some of our PIs on these studies, the reality is that everybody here works as a team. And I'm showing you the entire ADRC, all of whom have roles in many of the clinical trials and clinical studies that we do. So I'm going to stop there and I'm going to turn it back over to um, Dr. Sewell, and then we'll take some questions.
Great. So, Dr. Luizos, did you want to put up that question first or take questions first?
We can put the poll up first.
Okay.
So on your screen, a poll should have loaded. It'll have two questions. Just scroll down to the second question, click your answer, and then you can hit submit.
And then, so while everyone is doing that, let me just remind you, if you have questions, if you could please type them into the chat so that I can keep track of them. That would be great. So while people are still putting their responses in, Dr. Sano, let me start with a question that we get all the time in the ADRC, which is, can you just explain in plain English what a monoclonal antibody is?
Right. So this is a monoclonal antibodies are drugs that are developed to attach to a certain molecule or a certain protein in the body. So the monoclonal antibodies for amyloid attach to the amyloid protein. And the idea is that by attaching to it, it allows us to remove it. So this is a model, and the monoclonal antibodies are used in other diseases for other targets. But here, the monoclonal antibodies have been designed around the specific proteins of amyloid and parts of tau. And that the specific drugs can be different depending on what part of the molecule they attach to. And what I can tell you is that they are traditionally always delivered via either infusion or injection. They're not soluble, and so they don't cross the blood-brain barrier. And that's what makes their delivery more complicated than something that's oral.
So one question from the attendees are any of the studies you talked about applicable for people who might have moderate Alzheimer's disease, and how does mild and moderate kind of get
Yeah.
defined in these scenarios?
Yes, well, so it's a very good question. I would say that right now, most of our studies are defining themselves as being available for mild AD, or excuse me, or MCI, mild cognitive impairment. And when you think about more advanced cases, I think what's going on is that we are um, identifying people earlier, and our expectation is that's when we would treat. Now, what can be used in a more advanced case? We have the known treatments of the cholinesterase inhibitors and memantine. But I suspect as we get more comfortable with the use of these monoclonal antibodies, the possibility of introducing them to people who are somewhat more advanced, when we can have a better picture of their safety profile and their effectiveness profile, we may have a chance to expand them. At this point in time, we don't. But how do we define them? Typically, they're defined by either a mental status test, like a mini mental or a MoCA, or they can also be defined by the something called the CDR, the Clinical Dementia Rating, and that's a scale in which we look at both cognition and function. And if the function is not too seriously impaired, then they may actually be eligible.
Okay, great. So the next question is about potential dangers or side effects of the medications being used in these trials. I know there's been a lot of discussion around the new medications in the last few years that may have side effects. So the question is, what do we know about these ones?
Right. Well, first of all, anything that's given that's an antibody, or that's given either by infusion or injection, is likely to have a local site effect, right? So we know that already. That's a very uh, easy to identify possibility. However, that's a pretty treatable side effect. We have ways to treat local site uh, disturbances that we can treat pretty carefully. Right now, perhaps the most concerning one with the anti-amyloid treatments is what we call ARIA. And ARIA can be as minor as an evidence of a stroke that has only an image and no effect, um, or as serious as a cerebral bleed, a hemorrhage. So the range of the seriousness of this effect is broad, and we do know fairly well that certain individuals are more or less likely to have that effect, and that's specifically around the presence of the apolipoprotein E4 allele. Now, we believe that's particularly associated with the anti-amyloid agents. We don't know yet what the association of apolipoprotein E4 and tau will be. We just don't know if those antibodies will have the same interaction. But one of the things to keep in mind is individuals who have these one or two E4 alleles are more likely to have um, these silent looking infarcts in their brain. The ARIA is going to be there just by the likelihood of having the ApoE4 allele. And the treatments also require that the level of that kind of side effect that can pre-exist before you even had the treatment be low enough so that the added risk of the treatment doesn't put you in greater risk.
Thank you. Next question is, this is a great question. I'll just read what it says. You mentioned identifying AD earlier and staging. Are we getting to a point where we can identify and potentially treat or intervene before any cognitive symptoms, like someone with a strong genetic likelihood of getting AD? Currently, it seems like one has to wait until they have symptoms to get intervention.
It's a very good question. There are major trials going on right now with individuals who have amyloid and no symptoms to see if they can get a benefit out of removing amyloid with the monoclonal antibodies that are now available and have been approved by the FDA. It's an unknown question, as it's an unknown response here to say that it can remove the amyloid, but will it which I think is probably very likely, but will it reduce the likelihood of symptoms? That's what we don't know. And the risk with these agents is still high enough that if you weren't getting a clinical benefit, you'd really be unsure that it would be the right treatment.
Okay, good. So this next question has to do with sort of eligibility. They're saying, so now we can say a person has Alzheimer's if they have amyloid, even without any findings of MCI. And yet we're also saying that you have to have MCI to get into the trial.
Right.
So that's her
Okay,
question.
so it's become a very complex question. What we can say is that if you have amyloid, we can say you have Alzheimer's disease. That's the language that we use. Because you have amyloid, you have Alzheimer's disease. However, whether the amyloid is related to your symptoms at the moment, right? That depends on separately looking at your clinical profile. So your mild cognitive impairment is detected and separately from your detection of amyloid. In one common result that people may not appreciate is that most times when people come to us and say, oh, I don't have any symptoms, but I think I might have Alzheimer's disease, when you probe very carefully, they may have actually seen a symptom that they're not sure they can share as a symptom, a subtle subjective memory problem that we can't measure with our cognitive tests. That is possible. So that's the reason that we separate the two. But the presence of amyloid does not guarantee that you have either subjective or objective cognitive deficit. So it is true we can detect it earlier if we know to look earlier.
So this next question we get a lot. This particular question is, are participants in the AHEAD 4-5 trial ever able to switch to one of the other trials, or is it ever possible to do 2 trials of the ADRC? We get this question a lot with the ADNI study. They want to know if they can be in the ADNI study and also be in a trial. And this person specifically asking about AHEAD 4-5, either switching trials or doing 2.
So this is a really important question. And I think one of the wonderful things about the AHEAD trial is that they monitor your cognition as well as your amyloid throughout the trial. And if your cognition changes even slightly and they think that you've become MCI, they will actually offer the possibility to go on active drug. Now, it wouldn't make sense to go to another trial unless you knew what you were on in the first place. So I would say that AHEAD has such an evolved design that we can really assure patients that at the earliest time that we detect a change that puts you to the MCI diagnosis, we would offer to make a change for you and allow you to enter what we call the open trial. That means that you'd get the active drug.
Thank you. This next question is about for people already on a treatment. So the question is, if you have amyloid and you're taking the medication Leqembi clinically,
Mhm.
how would you decide whether to switch to the tau antibody rather than staying on Leqembi?
Right. Well, the tau antibody is in an early stage. It's called a phase 2 study, and they will not include anybody who's had any exposure to Leqembi. And the reason is because they're taking people at the same stage as Leqembi, and they want to find out if approaching the treatment of tau will be beneficial without even worrying about the amyloid. So for this clinical trial, only those who have had no exposure to anti-amyloid um, antibodies will be included. So you wouldn't be able to switch over. And it's not to exclude anybody, it's to get a reliable answer to the question. And if we the most important thing when we set up our inclusion-exclusion criteria is to make sure we can um, measure efficacy and answer the question as well as to determine safety. Those are the two factors that we use to see who we would exclude. And that's the reason these people would be excluded. We wouldn't be able to tell if the tau had a benefit.
Okay, okay. Um, yes, so just to make clear to everybody, the next question was some people came in a little bit late and they're wanting to know if they'll be able to watch this later. So absolutely, this will be up on our website in the next week or two, and you can certainly come back and watch if any parts that you missed. So the next question is a person with definite amyloid plaque but no MCI defined as having preclinical AD?
Yes, that was the term that we used to refer to as preclinical AD. And that term is still sometimes used. They also can be referred to as simply having Alzheimer's disease, but not Alzheimer's dementia or Alzheimer's cognitive impairment. But that is the term. Preclinical is just uh, a little confusing because in the research world, preclinical also means animal studies. And so that's why we've moved away from that preclinical term. But that is exactly what it is.
Yeah, that's the wording of all of that's been very confusing
Yes.
for many people. So another question was, how far along are we in our studies, and how soon will we have results for some of our trial participants?
Yeah. Well, the a head study and a study that is also being run called Trailblazer 2 or 3, I'm not sure. These are two studies that are moving along, and I believe next year will be telling us whether or not uh, there's an benefit of taking these drugs in asymptomatic people. The current studies that I've shared with you are primarily um, phase uh, phase 2 studies, which means this is developing the information to go into a phase 3 study. What's the kind of information that we're looking for? We're looking to see if it truly hits the target biologically. We're looking to see if it has a clinical benefit and where that clinical benefit might be so that we can design the phase 3 study to maximize. We're looking to see which dose is the best. So these current studies are a little bit further out because they're phase 2. But the timeline will depend on how much we get the information we need in the first set of trials. You can see most of these are 1 to 2 years plus recruitment time. So the results of these trials will probably be out in about 2.5 years and tell us what's next.
So we have to be patient a little bit.
Exactly.
It's hard. It's hard. This next question is, again, we talk about this all the time, a little bit about what's the relationship between the information you get from a PET scan that may say you have a lot of amyloid or not a lot of amyloid, and that what you get from a blood test. So can you be, quote, negative on a PET scan, but positive on a blood test, and how closely related
Yeah.
are those two methods of diagnosis?
Yes, those studies are being done, and many of them have been done, and they're incredibly accurate. The blood test is incredibly accurate. Let me tell you a little bit about where the difference might lie. So the amyloid tests for amyloid are becoming very good at predicting what your brain scan would say, particularly your amyloid brain scan. And one of the ways they do that is that their cut is created so that you have high confidence of positivity. And the ones that don't have positivity, there may be more of a range of people who would not be called positive. Although several of our studies have looked at even mild levels of change in the blood, of biomarker in the blood. So the blood test accuracy, if a blood test is positive, by whether it be clinically or in our research studies, we've seen very little disagreement with that result and the imaging studies. So the advantage, of course, is that we don't have to do quite as many PET scans, which are much more expensive and take much longer and use up more resource, particularly at the level of if you're A, going to treat, or B, you're trying to make a diagnosis. When we're doing research and we think we want to change the level of amyloid, we don't know that the blood test results is as accurate as measuring the change. And so for most of our clinical trials, if we want to confidently measure amyloid change, they will usually be doing an image.
That's interesting. A kind of related question from someone is, obviously, if we do a PET scan, we're doing it at this moment in time. And are you having enough amyloid in your brain at this moment to be considered for one of these studies? And the person asking the question wanted to know if they can be rescreened for a study if in the past their result was negative, but can that change over time? And does that mean that they could be screened for either that same study or a different study?
Yeah, that's a good question. I would say the interval is very important, right? So if the screening value, if a person was amyloid tested and it was um, considered not borderline, but simply negative, right? Repeating that scan in 2 to 3 years is the very soonest it should be repeated. Now, there can be a few exceptions. There's a belief that if you have a certain occurrences like head injury, you could have a more likely occurrence of amyloid. If your cognition changed dramatically and got suddenly worse, somebody might also recommend that. But I think that it's more likely they would recommend that you track with blood instead of with a scan, which is so resource intensive.
Okay. Okay. Another question is, at what point, if at all, do people find out what arm of a study they were
Yes.
in?
It's a very good question. So at the end of the study, we typically do expose or tell people, but it's not the end of you in the study, it's the end of the entire study. Because if we break the blind for one person, we actually reduce the blind in the overall study. So when the study is completed, and usually when the top line results are out, which means it has to be less than a year before the last person completes, I'm sorry, after the last person completes the last visit. That's typically when the sponsor will provide each of the sites with the information about what treatment a person was on.
Another person was asking specifically about the semaglutide study.
Yeah,
And
semaglutide. Yeah, very
yeah.
interesting study. So here's another example of
And they want to know when the findings, when we're going to
yeah,
be
I
findings.
know. The belief is that preliminaries will be out at the end of 2025, but the final results will be out at the end of 2026. This is a very large study, and one of the things that's interesting is while the company has been developing a story around how this drug might actually affect amyloid. In fact, the this class of drugs had its biggest effect in individuals who had diabetes and had some executive dysfunction, and it improved executive dysfunction. So it's really unclear. And this cohort that was recruited, they wanted to make sure that they could say it was Alzheimer's disease. So they all had amyloid, but they had very little diabetes because they were taking mild cases of Alzheimer's disease. So actually, there may not be a positive result because the cohort, even though they have Alzheimer's disease, may not have as many other comorbidities that could get a benefit from a drug like semaglutide. Alternately, maybe it has effects on these other biological systems that they're suggesting. So we'll have to wait and see.
Patience again.
Again.
A related question is, when are the top-line results for the AHEAD trial expected?
Oh, so that trial is 2 more years because while the recruiting is finished, it's a 2-year study, and the last people were recruited in October, I believe. And recruited then. I think some of their final visits were a little later than that, their first randomization visits. So that's at least 2 more years. So.
And I don't see any other questions in the chat. I know a question that we get a lot is, we've got a bunch of newish studies just opened in the ADRC, and how do you go about deciding like which one is best, or do you have an opinion about what's the best one to join?
Well, I try to tell everyone to pick the study that you can do. You've heard some of these studies are very intensive, right? They require monthly visits, they require infusions. Others are using a pill, right? Others are using a pill that doesn't exactly attack amyloid, but does other neuronal things. Each one of these studies will have very specific entry criteria. I've only given you the broadest strokes of the entry criteria, but they will have different concomitant medications that will be excluded. So one thing is to pick the one you're interested in and find out whether or not it matches your eligibility, and then go from there to think of the next ones. And our whole team knows all of these studies. And so they're able to share with you what order they think it's best in, not requiring, but allowing you to sort of think through the reasoning for each choice.
So in other words, if you don't get into the first study because you don't meet the eligibility, you could certainly be screened for other studies.
That's right. And the thing to keep in mind, however, is right now, all of our studies are for individuals who have Alzheimer's disease. That is, they have amyloid. So they could have MCI, but it has to be due to Alzheimer's disease. So
Except the ADNI trial, right? The ADNI trial is
right. It does. It has a cohort which will fill up quickly of individuals who can be normal, can have normal cognition. But in our other studies right now, we're really focusing on people who have maybe mild impairment, but have amyloid. And that's a very specific disease and a specific biology that we can now measure effectively.
Interesting. Okay, great. I think this is probably a good place to stop, but we've responded individually to some of the people who had questions that we might better address offline. If people have other questions, please feel free to email Dr. Luizos, who is the person who sent us this link, and she can figure out how to get the question answered. And again, this whole talk will be up on our website in the next week or two, if you would like to watch it again or watch from the beginning. So thank you, Dr. Sano, and thank you, everybody who joined us today. This was just a terrific talk. I really appreciate it.
Great. So glad I could be here.
Thanks so much. All right.